Welcome to the Deep Dive. We're here to take those really detailed academic studies and break them down into the key insights you can use. So today we're doing a deep dive on, well, an amphibian invasion, one that turned out to be much messier than people first thought.
Yeah, quite a tangled situation, actually.
Exactly. And our source here is this great paper in Heredity from 2025. It's by Everts and their colleagues. The title is Multiple Source Locations and Long-Distance Dispersal Explain the Rapid Spread of a Recent Amphibian Invasion. We're talking about the American bullfrog, Lithobates caspianus. Its rapid spread through the Grotonny River Valley over in Belgium. And the mission for you today is really to grasp why this wasn't just one simple invasion wave.
No, it was much more complex.
Right, like a chaotic multi-point blitz almost.
And that complexity really matters. I mean, if you're concerned about global biodiversity loss, invasive species are a huge piece of that puzzle.
Yes.
They drive decline through, predation, competition, spreading diseases. So for regional management, for actually doing something about it, you need to know the specifics. How exactly is this species moving? Simple assumptions just don't cut it.
And the simple assumption here, the sort of conventional wisdom about this bullfrog invasion.
Yeah.
What was that?
People basically thought it was straightforward. One primary introduction event, probably downstream from somewhere specific.
Creating one big metapopulation.
Exactly. And then spreading predictably, just following the river current downstream. Nice and neat.
But that neat picture started to fray a bit, didn't it?
It did. The evidence wasn't quite lining up. The spread seemed too fast, maybe too widespread for just one starting point following the river.
So they suspected something more complex.
Right. Multiple introductions, maybe happening at different times, asynchronous introductions, and dispersal that wasn't just downstream, maybe bidirectional, maybe overland.
Okay, so a much messier reality. How did they go about proving that? How do you untangle something like that?
That's the core of the study. They realized they needed to combine different kinds of evidence. Decades of field observations plus modern genetic analysis.
Right, that combined approach. Let's dig into that synergy because it sounds crucial. Why not just rely on, say, the field sightings alone? Or just the genetics?
Well, each one only gives you part of the story, you see. Genetics tells you about relationships, who's related to whom, but not necessarily the when and where the invasion started or the exact path it took.
Okay.
And the occurrence records, the sightings, they tell you where frogs are and when they appeared, but not how different populations are connected genetically or if they came from the same original source.
So you need both to see the full picture, how the genetics map onto the spread across space and time.
Exactly. You need that combination for real insight.
So let's talk data. What kind of occurrence records are we talking about?
A lot of data, actually. Nearly 3 decades worth. Collected mostly through citizen science. Yeah, over 1,500 sightings between 1997 and 2023. But they focus particularly on a subset of expert-verified records from before 2009.
Before 2009, why that cutoff? What happened in 2009?
That's when a legal ban on trading or possessing bullfrogs came into effect in the region.
Ah, okay.
So by focusing on those pre-ban sightings, they could be more confident that they were looking at patterns driven by the initial introductions likely from fish farms or the pet trade rather than just, frogs naturally hopping around later on.
Gotcha. So that helps pinpoint the likely origins. And the genetics. How many frogs, how many locations?
They got genetic data from 278 bullfrogs. These were sampled across 31 different spots covering the whole invaded range.
And what were they looking for in the DNA?
They analyzed thousands of SNPs, single nucleotide polymorphisms.
Okay, SNPs. Remind us what those are.
They're just tiny single-letter differences in the DNA code between individuals. Think of them like unique genetic markers or tags. They let you see patterns of relatedness and population structure. They looked at over 8,500 of them.
Okay, so a huge amount of genetic detail. Now they've got sightings over time and detailed genetic tags. What tools do they use to put it all together? How do you find those hidden starting points?
For the where, they used a technique called geoprofiling, specifically a Bayesian algorithm, the Dirichlet process mixture model.
Sounds complex. What does it do, basically?
Think of it like criminal geographic profiling, but for invasive species. It takes all the locations where the frogs were found and works backward to statistically estimate the most likely origin points, the hot spots where the invasion probably kicked off. They tested models for up to 6 potential source locations.
Okay, so geoprofiling estimates the where. Then how do they figure out who were these frogs all from one big family or multiple distinct groups?
That's where the genetic analysis comes in, specifically a method called structure analysis.
That's structure, right?
It looks at all those SNP markers across all the individuals and figures out how they cluster genetically. Does everyone belong to one big mixed group, or are there several distinct genetic populations?
And they also looked at the landscape itself. You mentioned mapping resistance.
Yeah, they used another tool called ResDisMapper. It basically overlays the genetic information onto the landscape features.
To see what helps or hinders spread.
Exactly. It helps identify things like canals, roads, maybe even elevation that act as barriers, slowing down or stopping dispersal. And conversely, it can show corridors, like the river itself, that make movement easier. It maps out the resistance or lack thereof to gene flow across the landscape.
Okay, so they've got the tools, they've got the data. What did they find? Did it support that simple single source idea?
Not at all. This is where that tidy myth really falls apart. The geoprofiling results strongly suggested not one, but 6 potential source locations.
6? Wow, that's a big difference from 1.
A huge difference. And the timing of the earliest sightings supported this idea of multiple starting points.
How so?
They saw 2 distinct initial clusters emerge. First, a northeastern cluster, or NE, popped up around 1997. That one seemed linked to a fish farm nearby, a likely pathway.
Okay, that's one potential origin.
Then a few years later, around 2001, a separate southwestern cluster, the SW cluster, was first observed. This one was near an area with cottages and ponds, suggesting maybe escaped or released pets were the source there.
So different times, different likely reasons for introduction. But wait, you said the geoprofiling pointed to 6 source locations, but then the genetic analysis.
Yeah.
What did structure show?
That's the really fascinating part. Despite the geoprofiling suggesting 6 potential geographic origins, the structure analysis only found 2 major genetic clusters.
Only 2? How does that work? 6 locations, but only 2 genetic types.
Exactly. That's the key nuance. It suggests that there were at least 2 primary introductions from genetically distinct source populations corresponding to those NE and SW clusters.
Okay.
But the other 4 locations identified by geoprofiling, they were likely founded either by secondary introductions, maybe people moving frogs from the initial spots using frogs that were already genetically similar, or through natural long-distance dispersal events, LDD, where frogs from one of the main clusters managed to jump further out and start a new population, but they carried the same basic genetic signature.
I see. So not necessarily 6 independent founding events from unique sources, but maybe 2 primary ones, and then subsequent spreading or moving of those same genetic types.
Precisely. And the landscape played a huge role in maintaining this pattern.
The ResDisMapper results.
Yes. It clearly showed that a major canal crossing the river valley acted as a significant dispersal barrier.
So it physically separated them.
It really did. It seems to be largely responsible for keeping those NE and SW genetic clusters spatially distinct, even though they're relatively close geographically. Gene flow across the canal was much, much lower than along either side of it.
Okay, so we have multiple sources, 2 main genetic groups separated by a canal. But the overall spread was still fast, right? What did they find about the speed and pattern of the invasion front?
It was fast, yes. The main expansion was downstream along the river, which you'd expect. But the interesting thing was the rate. It expanded at a pretty consistent linear rate.
Linear, not exponential.
Mostly linear, yeah. Averaging about 1.45 kilometers per year overall. Often with invasions, you see an initial exponential boom phase, but here it was more of a steady linear advance.
And was the speed the same everywhere?
No, there was a difference between the 2 main clusters. The southwestern group, the one potentially linked to the pet trade, that one actually spread faster, about 818 meters per year on average, compared to the northeastern cluster, which advanced at about 631 meters per year.
So the SW cluster was moving quicker.
Yeah.
Now, what about the direction? Downstream expansion makes sense for river species, but was that the whole story for gene flow?
That's another fascinating finding. While the range expanded mainly downstream, the genetic analysis showed evidence of gene flow happening in both directions, upstream and downstream.
Upstream, too. How does that happen if they're mainly following the current?
That's the question. It strongly suggests that other things are going on besides just passive drift with the water, maybe significant overland movement. Frogs hopping across land between ponds or ditches.
Oh, people moving them.
Or potentially ongoing, perhaps accidental, human-mediated dispersal. Secondary introductions, moving frogs against the current, or just between unconnected water bodies, it breaks that simple downstream-only rule.
And this complexity gets amplified by environmental factors too, right? You mentioned long-distance dispersal, LDD.
Yes, exactly. They found evidence suggesting that major flood events played a role.
Flooding. How?
Well, near one of the source locations, location 3, they observed that periods of extreme rain and flooding seemed to coincide with the establishment of new populations much further downstream locations 5 and 6.
So the floodwaters just wash them downstream.
It seems likely floods can act as this massive passive dispersal mechanism, carrying eggs and tadpoles way beyond the normal range, effectively jump-starting these satellite populations. That's a classic LDD mechanism in river systems.
Okay, so you've got multiple introductions, bidirectional gene flow, landscape barriers, and these big flood-driven jumps. What does all that mixing and moving do to the genetics over distance? Did they see the typical pattern of diversity loss?
That's another key point. They didn't see strong evidence for what's called serial founder effect.
Serial founder effects.
Yeah.
That's when populations lose genetic diversity the further they get from the original source, right?
Yeah.
Like each new step involves only a few founders.
Exactly. You typically expect heterozygosity, a measure of genetic diversity, to decrease with the distance from the source in a simple stepping stone expansion.
But they didn't find that here.
No.
Yeah.
There was no significant correlation between how genetically diverse a population was, its heterozygosity, and how far it was from the likely source points identified by geoprofiling.
Why not?
Because of all the complexity we've just discussed, the multiple introductions, the constant mixing from bidirectional gene flow, those LDD events, reseeding areas, it all works against that steady loss of diversity. It keeps the gene pool stirred up across the invaded range.
So high genetic diversity is maintained throughout the invasion front.
It seems so, which likely contributes to their success. They aren't suffering from inbreeding depression or lack of adaptability that you might see in populations founded by just a few individuals. This lack of strong founder effects means those classic source-sink dynamics aren't really the main story here. It's much more mixed up.
Okay, so understanding this complex web of introductions, barriers, and dispersal, what does it mean for actually managing these bullfrogs?
It has pretty direct implications. For instance, we now know that the northeastern cluster is kind of boxed in by canals upstream and downstream.
Right. The ResDis mapper showed that.
So that population might reach its carrying capacity, the saturation stage, sooner than the other one. Management efforts there, like removal programs, might be more contained and potentially more effective in the long run.
Whereas the southwestern cluster, the faster spreading one,
That one has fewer obvious barriers downstream and seems to benefit from those flood events spreading it passively, so it's likely to just continue expanding along the river valley. Containing that one will be a much bigger challenge.
it really highlights how understanding the specific local dynamics is crucial for planning.
Absolutely. And the study makes it clear that the rapid spread isn't just about the frogs themselves, but about this high degree of genetic mixing, the multiple starting points, and the landscape interactions. The post-introduction dispersal is really key.
Did the authors acknowledge any limitations? It sounds like they untangled a lot, but were there still uncertainties?
Oh, definitely. They're upfront about the challenge of using genetics alone to perfectly distinguish between, say, a natural long-distance dispersal event by floodwaters versus someone moving a few frogs in a bucket, especially if those frogs came from the same original genetic cluster.
Right, the genetic signature might look similar even if the mechanism was different.
Exactly. That remains a tricky area in invasion biology. But even with that caveat, the main conclusion is robust. That the simple, single introduction, downstream-only model is clearly wrong for this invasion. It's undeniably a process driven by multiple asynchronous introductions, amplified by LDD events, and significantly shaped by landscape features, especially man-made ones like canals.
So the takeaway for you, our listener, is that this bullfrog invasion in Belgium wasn't a neat wave. Think of it more as a messy, multi-front advance, fueled by genetic mixing from different starts, helped along by floods, and channeled or blocked by things like canals.
Much more complicated, right?
Much more. And looking forward, the authors really champion this combined approach, using occurrence records and stressing the huge value of citizen science data there alongside detailed population genetics. That's the powerful toolkit we need to track and manage invasions like this effectively. It gives you the where, the when, and the how.
Okay, that leads nicely into a final thought for you to chew on. Remember that legal ban on trading and possessing bullfrogs back in 2009?
Well, this study found evidence for ongoing bidirectional gene flow, which, as we discussed, might partly reflect continued human-mediated spread even after that ban. So what does that suggest about the lingering, maybe hidden influence of things like the pet trade even years after regulations are put in place.
Oh.
How persistent are these pathways?
That's a really important question, with implications far beyond bullfrogs in Belgium.
Something to think about. We'll leave it there for this deep dive.